Okay, so we're back to our test 5 review. Drop frames detected, 30 frames per second. Okay, YouTube thing is surely complicated. Anyway, so apology for just now. The computer was crashed while I was talking about insulin and blood glucose level. So I guess it acts as a good break for our long review. So let's just wait a few more seconds to see who will be joining us for our today's review. So test 5, as usual, will be covering biological organizations, atoms and molecules, biomolecules, a tour of the cells, protein synthesis, human system and tissues, and homeostasis. If you look at the notes, if you go to the notebook, we already have a few questions answered. So first is that what are the 10 systems in the human body? We have 10 of them, and if you wanted to add integumentary system, then there will be 11. We have endocrine system, nervous system, lymphatic system, skeletal system, digestive system, reproductive system, muscular system, circulatory system, urinary system, and respiratory system. And each system has its own functions. So you need to know which systems do what. You need to know how do systems work together in order to maintain the body's homeostasis or internal balance, and be able to know which organs, at least some general idea on what organs corresponds to what system. Muscle as an organ that is in muscular system. Heart is made out of muscular tissue, but it is involved in circulatory system because it pumps blood. You also have the stomach, which is in the digestive systems. So be able to know by looking at the descriptions of the organ, you know what system is under. Some organs can be in different systems, and different systems can work together to perform one specific task. Like for instance, if you wanted to get oxygen into the body, you need circulatory system as well as respiratory system to work together. Respiratory system gets the oxygen into the body, and the circulatory system transport the oxygen to every parts of the body. So that's the big idea. System works together. Systems have different organs working together. Organs are different tissues working together. And tissues are a group of cells performing similar tasks. Next is the 4 types of human tissues: epithelial, connective, nervous, and muscular tissues. Know what are the functions and how do they look like. Hi Cheyenne. You missed out our first part 1 review session, so now this is live part 2 because my computer crashed in the middle of our review session, so too bad. Okay, but that's good. It acts as a good break in between because I did not know that our review is gonna last for such a long hours. So be able to identify the different types of tissues, what are their functions, and what jobs do they do. Next, be able to understand homeostasis, how different organs work together so that your body's factors or variables are always within a certain range. So we have homeostasis, which means maintaining the internal body balance. In this example, you can see that it is about it's about temperature, right? Body temperature. If temperature increase, I'm not sure if you can see the mouse, but if temperature increase, you can see that the brain is going to receive some sort of signal, tells the sweat gland and blood vessels to dilate so that it does something to decrease the body temperature. Decreasing body temperature when the body temperature is increasing is a way to maintain homeostasis. You can see that they are opposite, right? When temperature increase, the brain sends signal to decrease the temperature. So we call this negative feedback. The body does something as a counteraction to negate or counteract, do the opposite of what is changing. If temperature is increasing, the body is going to do something so that it can decrease the temperature, which is the opposite of increasing. So that's negative feedback. And as you can see, when the temperature is decreasing, right? Just now it was increasing, so the body does something to decrease the temperature, so the temperature is going to start decreasing, but you don't want the temperature to drop so much to the extent that you become like an ice. So when the body detects that the temperature is starting to drop and is going to drop below the normal range, the body is going to start telling the body to stop the cooling mechanism. So it's going to stop the body from continuously cooling down, right? So this is also an example of negative feedback. The body is cooling down because of the cooling mechanism. So the body is going to do the opposite. Instead of continuing the cooling mechanism, it's going to stop the cooling mechanism. So if you can see, this ends up being a loop, a circular loop, increasing the temperature. The temperatures increase. The body is going to do something to decrease the temperature. So the temperature is going to start decreasing back to normal. A loop is going to be the same as when temperature is decreasing, when you're in a very cold weather right now, like this snowy day. Okay, so feel free to shout out. What do you do today during the winter, like this snowy day? Do you drink chocolate drink? Let me know what's your favorite drink when you are in this very cold weather. I exceptionally love drinking Milo, so it's a chocolate drink, hot and warm. Okay, and also understand about positive feedback. So now we're going to go into the glucose, right? How to use insulin to regulate blood glucose level. Mr. Wong's favorite food is ice cream. So when I'm stressed, I would love to eat ice cream. So when I eat an ice cream is full of carbs and it's full of sugar, it's very sweet. And when you eat ice cream, it's going to increase your blood glucose level, which means in the blood vessels, there are a lot of glucose. So glucose in our picture here is in green, right? When you eat ice cream, blood glucose level is going to increase in the blood vessel. So blood vessel is the tube that carries the blood. You don't want the blood to have full of glucose because it's going to make you diabetic. Diabetic basically means that there's a high blood glucose level in the blood vessels, but higher than average people. And it's going to cause a lot of problem because if you cut yourself, your blood is going to be very sweet, high in glucose, and it's going to be a huge attraction for fungi infections and yeast infections because they also thrive on glucose to grow. So they want those glucose as a source of energy. So you don't want that to happen. So what happens is the body is going to start decreasing the blood glucose level when there's an increase in blood glucose level. Negative feedback, remember? Negative feedback. So pancreas cells, pancreas is an organ in your bodies. It's part of the digestive system, it's also part of the endocrine system. It's part of the digestive system because it helps regulating intake of nutrients. It's also part of the endocrine system because it releases hormones into the body. So the hormones that the pancreas is going to make, one is insulin and the second one is glucagon. Insulin is going to decrease blood glucose level, while glucagon is going to increase the blood glucose level. Okay, so when there's a high blood glucose level in the blood vessels, insulin is going to get released by the pancreas into the blood vessels. So insulin is going to get released. So I'm going to draw insulin as black triangles. And insulin is a type of hormone. It's a protein hormone. It's going to bind to cells, like for example, muscle cells. So I'm going to draw a muscle cell at the downstream of the blood vessels. What it means is that it is, you can imagine the blood as like a river, upstream, downstream. Blood flows in one direction. And muscles is below where the pancreas is. So pancreas is going to release the insulin into the blood vessel. Blood is going to carry, like a river, carry the water. Insulin is going to start flowing in one direction until it reaches the cells where it needs to be. And muscle cells has receptors on it. Receptors is where protein binds. So protein, the insulin is a type of protein, is going to bind to the receptor. So this is receptor. A receptor is going to tell the muscles to take in the glucose into the muscle. So this cells is going to go, this glucose is going to move into the cell, because this insulin receptor and receptor binds to the insulin. Insulin is going to tell the receptor to tell the cell to make more channel protein. Right? We remember that every cell has a membrane. Membrane is going to prevent things from going in and out of the cell. And glucose is kind of big to go in or out of the membrane. So we need some sort of transport protein. Remember what we learned about the cell? Cell has transport protein embedded in the phospholipid bilayer of the membrane so that they can move things across the membrane. Only small and non-polar things can move through. Occasionally water, but even because water is very small and water is polar. So sometimes water can be moved through the membrane, but it's not the most effective way of moving across the membrane. And glucose, non-polar, and it's ridiculously big. So you cannot just simply squeeze the glucose through a cell. You need to use special protein that transport the glucose into the cell. That's why with insulin. Insulin is going to tell the cell to open the gate, and the gate is the channel protein. And the protein is going to move, the glucose is going to move into the cell through the channel protein. So let me repeat that again. The channel protein, right? The channel protein act as a gate, channel protein act as a gate so that it allows the glucose to move into the cell, the muscle cell here. So this is muscle cell. So why specifically muscle cells, right? We know that muscle involves in a lot of movement and it burns a lot of calories. So what does it mean when we say a human or a person goes work out, uses muscle, burn energy? Because muscle is very rich in a type of organelle called mitochondria. Right? If you remember, mitochondria is a powerhouse of a cell. So let me just draw a nucleus of this cell so that you know that this is a cell, a muscle cell. So mitochondria is the powerhouse of the cell. It makes ATP. So how does it make ATP? We learned it from previously that it can turn glucose with a little bit of oxygen. They're going to turn it into carbon dioxide and water. This is respiration, right? In this process, it's going to produce a lot of ATP, and ATP is what we call the currency for energy. It stores energy in this molecule. ATP is a type of molecule that carries energy. So we have muscle cell that can turn glucose, right? So all this green thing that we just absorb from the blood vessels, they're going to turn them into lots of ATP. So what if you are a very lazy person, you don't work out a lot? The muscle is still going, or the cells still going to take in a lot of glucose. Instead of turning them into ATP, they're going to turn it into storage of energy, right? Because they're not using them. You don't do a lot of workout, you don't exercise a lot, so you don't use ATP, you don't use a lot of energy. So they're just going to store the energy as storage, so that when you need to use it, you can turn them back into energy again. So the muscle cells can either turn glucose into ATP, or they turn it into glycogen, right? So glucose to glycogen. And if you remember, glycogen, from our biomolecule chapter, glycogen is a type of polysaccharides. if you remember our previous chapter, we learned that polysaccharide is a huge molecule made out of monosaccharides. So a lot of monosaccharides binded together through dehydration synthesis, right? We form a bond between two monosaccharides so that they can become disaccharides or polysaccharides. They're bonded together. So this is a way of storing energy. We take the cells taking a lot of glucose, bond them together, and make them into glycogen. Then it becomes a storage. And surprisingly, glycogen is not sweet, and it can change a lot of glucose, store them, like 10,000s of them, into glycogen so that it doesn't affect your blood glucose level. The glucose is not in the blood anymore. It's now in the cell. It's been stored away from the blood vessels. So you can imagine that only when the cell needs energy, then they will break the glycogen, right? When the cell needs energy and you didn't eat a lot of things. So when you did not eat a lot of things, your blood glucose level is very low because there's not enough glucose in your blood. You don't eat. So your cell needs glucose now, but there's no glucose in the blood vessel. So they're just going to break down glycogen into glucose, which is a type of monosaccharides. That can be used by mitochondria to make ATP. So the process of turning glycogen to glucose is hydrolysis, the breakdown of a polymer to a monomer. Glucose is a monomer, glycogen is a polymer. So the way how they turn it into glucose from glycogen is the use of hormone glucagon. So know the difference between glucagon and insulin. When your blood glucose level is very high, which means that you just eat a lot of food and your blood has a lot of sugar. And we don't want that. There's an increase in blood glucose level, so we need to transport the glucose from the blood vessels into the cells by using insulin. So insulin is going to secrete into the cell from the pancreatic cells, bind to the receptor of the cell. The cell is going to make a lot of channel protein, and glucose is going to get sent into the cell. So what if there's not enough glucose in the blood vessels? They're going to release. Okay, so when let's say you starve for like 5 to 6 days, you haven't eaten for a long time, you need a lot of, there isn't a lot of glucose in your blood vessels, and the muscle has used out all the glucose available, so there is not enough glucose. Your body is going to secrete glucagon. Glucagon is going to go into the cell, right? Go into the blood vessels, and then glucagon bind to the cell is going to tell the cell to break down the glycogen. It's going to tell the cell to break down the glycogen that is stored in the cell, so glycogen. I'm going to break down glycogen into glucose, and then glucose is going to get released out of the body. And that's how you use insulin and glucagon to regulate blood glucose level. And insulin and glucagon, they're both protein. So they use the transcription, translation, and all the organelles in the cell to make them. Next, you need to learn how to tie everything that we have learned so far into homeostasis. We learn about cell. Cell has a membrane. And if this is a eukaryotic cell, it has a nucleus that stores the DNA or genetic material. All types of cells have membrane and cytoplasm and genetic material. So cytoplasm is the region between nucleus and membrane for eukaryotic cells, and if you are a prokaryotic cells, you still have membrane, you still have cytoplasm, except your genetic material is just kind of flowing around in the cell without a nucleus to store them. So there's a difference between prokaryotic cells and eukaryotic cells. Prokaryotic cells, pro means before, you don't have a nucleus. And eukaryotic cells, eu means new or real, you have a new nucleus, real nucleus. Next, this is a membrane. So if you remember about the mascot chapter, I asked you to draw and design a mascot of phospholipid. Phospholipid has a hydrophilic head and a hydrophobic tail. Hydrophobic tail has to bond to hydrophobic tail. So a membrane always have head and head lining up and tails getting stuck in the middle. So that's the structure of a phospholipid bilayer, which is the structure of your membrane. And if you keep repeating this process, you're going to make a cell that differentiates between what's inside the cell and what's outside of the cell. And the nucleus stores the genetic material. And we know that the molecule that stores the genetic material is the DNA. DNA is double helix. Some of the DNA are single helix, single strand, but general rule, at least for human, double helix, which means it has two strands. The bases are opposite. If you remember, A binds to T, A bonds to T, and C bonds to G. Okay? So, the bonds between the bases are hydrogen bonds, which means they have attractions between the bonds, between the bases, and you can open up them or close them together. Depends on activation energy. You can use protein to open them up or close them up. That will be a little bit later in future chapters about how DNA opens up or closes down. But the idea is that DNA is in the nucleus, and they carries the genetic material or the genes that codes for your traits, right? The features that you need, that you can see from the outside. And these features are determined by the proteins that you have. Let's say if you have dark skin, you have a type of protein that makes your skin dark. If your skin is fairer or lighter, it means that you do not have that protein to make your skin fairer or darker, right? Same goes to insulin and glucagon. People with diabetes means that they do not have insulin protein. The cells have some sort of sickness or disease. We call them mutation, some sort of changes that makes them unable to make the protein. So they cannot make the protein, which is the insulin. So they cannot make the insulin and send it out of the cell so that they can turn glucose into glycogen. That's why their blood glucose levels are so high. They don't have insulin. Of course, there are many different types of diabetic patients. We're focusing on type 1 diabetes, people who are naturally born with this sickness. Okay, so now we have learned about how a cell makes a protein already. We learned about the different organelles that makes protein or involves in the protein synthesis process, right? Making protein synthesis. Synthesis means making. DNA is in the nucleus. DNA carries a gene, so the code that tells the cell what to make, what proteins to make. And the machinery or the organelle that makes protein is called ribosome. So ribosome is an organelle that makes protein, but ribosome is outside of the nucleus, it's in the cytoplasm, it's not in the nucleus. So ribosome doesn't have the information to make the protein. So we have a messenger in between the nucleus and the ribosomes. The messenger is mRNA. So mRNA is the message that the nucleus or the cell has made based on the DNA. So based on DNA, they make a message called mRNA. They're complementary base pairs, so it means that they are oppositely based. If you have an adenine, you have a uracil. Because RNA doesn't have thymine, so change it to uracil. If you have cytosine, you're going to make a guanine. So you're going to have an mRNA. So based on what DNA you have in the nucleus, you're going to start making an mRNA. So this mRNA or messenger is single-stranded because it's RNA. RNAs are mostly single-stranded. So it's small enough to move out of the nucleus. So mRNA is going to move out of the nucleus. The whole process of making this mRNA based on the DNA is what we call transcription. Transcription. Making a script, and it has a C. So C goes before L. So it's the first thing that happens in the protein synthesis process, making a script based on the DNA, and the script is mRNA, the message. So mRNA is going to move out of the nucleus into the cytoplasm where it can meet ribosome. So mRNA meets the ribosomes. So ribosome is going to use this mRNA to make protein. The way they make protein is to use tRNA. tRNA stands for transfer RNA, and it usually looks like a T shape. It has an amino acid on top of it. If you remember, the basic building block of a protein is amino acid. So if I want to make protein, I need amino acid. So someone has to bring me in amino acid. So tRNA is going to bring the amino acids in the cell to the mRNA at the ribosomes. So how do they know what amino acids to bring in then? There are 20 different types of amino acids. Each mRNA has a code in there. We call it codon. It's a 3-base code. 3 bases code. So they're going to read it every 3 bases. So like AGG, it will be translated into one amino acid. So the code on the mRNA is called codon, and the code on the tRNA is what we call an anticodon. So it's basically the opposite. They're going to match with the codons. That's why we call it anticodon. Anti means matching, going against, matching. So amino acid on the tRNA, based on the anticodon on the tRNA, is going to match with the mRNA's codon, going to match with the codon on the mRNA, and then they're going to start bonding the amino acid. So the amino acid in the ribosome is going to start making bond. And the process of bonding amino acids is dehydration synthesis. Right? Synthesis. We have been repeating this over and over and over and over again. Whenever we are in the new chapter, we're still using the previous chapter's material. So that's why I emphasize a lot on the continuations, right? Starting from the first lessons, we're going to repeat the first lessons materials, lessons that we learned from the first semester. We're just going to keep talking and talking and talking and talking about the same material and apply it in biology. And if we link amino acids together, you're going to form bonds. The bonds between 2 amino acids is a peptide bond, PEP. When you say PEP, it is protein, something related to protein. So peptide, protein bond. We're making protein. Building block, amino acid. So once the ribosomes finish making the protein, oh, by the way, the RNA that makes the ribosome is what we call rRNA. If you remember, we learned there are 3 types of RNA, the RNA, tRNA, and mRNA. The process of making this protein is what we call translation. We're translating DNA into protein. There are 2 different molecules, 2 different languages. Nucleic acid is made of nucleotide, and we're translating nucleotide into amino acid, 2 different languages. That's why we call it translation. And it's also L. L goes after C, so transcription happens first, then translation, right? So C and then L. So the process of making protein from the mRNA is what we call translation. So once ribosome has finished making the protein, it's going to get shaped in the endoplasmic reticulum. If you remember, we learned there are 2 types of endoplasmic reticulum. The first endoplasmic reticulum is a rough endoplasmic reticulum. This rough endoplasmic reticulum has some unique features on it that makes it rough, which is the ribosomes. So we remember, ribosomes makes protein. So ribosomes, so this ER is rough ER. So ribosome makes protein. So when you finish making the protein, it's still not a functional protein because it's just a strain of amino acids, right? We call it polypeptide. A lot of amino acids bonded together. Then rough ER, if you remember from a tour in the cell, we learned that rough ER involves in transport of protein. So protein is going to get shaped in the rough ER. So the ER acts as a highway to move the polypeptide to places where it needs to be. Rough ER then sends the protein out to Golgi. So let me finish drawing a Golgi. So Golgi, let's just write it like here. So it's going to send the protein to Golgi. Golgi is like a post office. Golgi will take in the polypeptide made by the ribosome, shipped by the rough ER. Golgi is going to package it, right? Fold the protein. They take in the protein, and then they start folding it, packaging it, and secrete it. What it means is that Golgi is like a post office. They look at the protein, be like, maybe I can make it a little bit prettier, a little bit more 3D. I'm gonna fold it into different shapes. So they're gonna start folding it, make the protein a little bit nicer, a little bit more 3D, so that it can be used, because protein is to be in shape, so that they have the active site in order to bond to something, right? To bind to the substrate. Protein. So protein get folded by Golgi. Golgi is gonna make some modification and then send it out. So the protein is eventually going to be folded with some sort of shape. So I'm gonna draw something like this, a shape, and it's gonna get shipped out in a vesicle. And then the protein is gonna get released out of the cell. And in our case, it is the insulin that we're making, right? So the insulin is the protein that is being made by the cell and is being shipped out of the cell. And that's the whole process. And that's why we're learning so many things from the very first chapter. If we remember, from our first chapter, we learned that everything is made out of atom. And then atoms group together, bonded together, and become molecule. And then different molecules bonded together, not bonded together, they form together to form a special parts of the cell, which is organelle. Then organelles is going to make a cell. Cell is made out of different organelles. Then cells, a group of cells, is going to become tissue. Different tissues work together is going to become an organ. Different organs work together is going to become an organ system. A different system works together to make a human or an organism. So we learned that atoms is the basic unit of matter and is made up of 3 subatomic particles: proton, neutron, and electron. We learn about their charges and how they work together so that you can have one building block of stuff, matter, right? Then just one single drop of water has many different atoms in it. So we learn about how atoms can connect with each other to form a molecule. So a molecule is a group of atoms bonded together either through covalent bond, ionic bond, or even hydrogen bond. Hydrogen bond is a type of intermolecular bond. So it means it's the bonds between 2 molecules that attract to each other because they are of different charges, right? The charge is due to electrons and proton. So we learned there are different types of molecules. We learned about oxygen molecule, which is the oxygen gas, the gas. We also learned about water molecule, right? Water, hydrogen, hydrogen, and oxygen. It is the polar molecule, so it can connect to each other. Covalent bond and ionic bond are what we call an intramolecular bond. So basically they are bonds in between atoms that makes a molecule. So covalent bond is formed through sharing of electron. Ionic bond is formed through transfer of electrons so that you can make ions. Right? Ionic bond. covalent sharing of valence shell's electron. So now, learning about atoms, we know we can make different molecules, but they are still not living organisms. These molecules are not alive, right? We learn about how we can know what molecules is alive and what molecules not alive. There's some molecules, and we have 7 criteria to determine whether something is a living organism or not, right? The seven criteria are patterns, evolutions, adaptations, regulations, response to external stimuli, growth and development, and I think that's all. Yeah. So you see that we learn about the criteria so that we know what's alive and what's not alive. And we know that at this level, it is still atomic level, so they're still not alive, they're inorganic. And now we're going to learn about organic. We learn about different types of organic molecules, right? Molecules that are important for living organisms to be alive. So we learned the 4 types of different molecules that are essential for living organisms: the carbohydrates, the lipid, the nucleic acid, and protein. So there are 4 different types of biomolecules that are essentials for us to be alive. Carbohydrates, nucleic acid, from the process of this covalent bond, hydrogen bond, how they interact with each other because of their functional groups. Next, by using these different types of molecules, you can make organelle, right? The different parts of a cell. So you learn about the different parts of a cell. For instance, the plasma membrane, which is made out of a molecule that is called phospholipid bilayer. So phospholipid bilayer is made out of phospholipid. You have the hydrophobic tail and hydrophilic head. If you have a lot of them, essentially you're going to make a membrane that separates what's inside the cell and what's outside the cell. You also learn different kinds of organelles, right? And all these organelles are made out of molecules. Depends on what organelles they are, they have different components of biomolecules. Then you have cell, different types of cell. We learned that prokaryotic cells and eukaryotic cells. And eukaryotic cells. The difference is nucleus. Eukaryotic has nucleus. And we learned about taxonomy before. Prokaryotes are bacteria and eukarya. They are only one single cells and they're still alive, but they do not have a nucleus. Their genetic material is not stored inside the nucleus. While eukaryotic cells are under the domain of eukarya, and all eukaryotes kingdom have eukaryotic cells. And you learn there are 4 types of them: the fungi, protista, plantae, plants, and animalia. Basically, fungi cell, protist cells, plant cell, and animal cells, the 4 major type of eukaryotic cells that make up the eukarya domain. So you have plants and animals. Then we learn about the differences between plants and animals. Plants have cell wall, which is made up of a molecule called cellulose, which is a type of carbohydrates, and animal cells does not have cell wall. Right? And I believe Mr. T also mentioned about the Endosymbiotic Theory. Is the idea that eukaryotic cells are kind of like 2 different types of prokaryotic cells being eaten or being living together inside of each other, and they kind of live happily ever after. So now they give rise to all the different types of eukaryotic cells. But that's not important in our coming test. The idea is that molecules gives rise of the organelle, and different types of organelles makes up cells, and different types of cells have different types of organelles that mix up with different types of molecules. That's why we learn about molecules, we learn about atoms, we learn about polarity, so that we know how they interact with each other in a cell. Then we learn about different types of tissues, the 4 types of tissues, which is epithelial tissue, muscular tissue, connective tissue, and nervous tissue. And these are basically groups of cells working together for one specific functions. Then different tissues work together to make organs. So we have different organs. We learn about heart, brain, kidney, pancreas, so on and so forth. Then we learn how these organs work together in different organ systems, right? We learn about endocrine system, and then nervous system, circulatory system, so on and so forth. And then this different system can helps an organism, so basically you as a human, how this organ system works together. We learn how this organ system works together to maintain homeostasis, make sure that your internal balance stays there. Homeostasis is also a type of criteria for living organism, right? If you are a living organism, if you're alive, you have the capability to maintain what's inside of your body. So us human, we're alive, we can maintain our internal balance automatically. Homeostasis. We learn about how to regulate temperature. We learn about how to maintain our glucose level. By using this different organ system that use different organs, different types of tissues, different types of cells, different types of organelle, and how they work together to release different kinds of molecule made out of different atoms, so that you are alive. And that's the major objective of what we're learning so far, and it's all tied together. And by the end of homeostasis chapter, you have a good understanding of how these cells can actually be so important in regulating our body so that we can always be healthy. We walk around, we exercise because we wanted to burn our glucose, make it into ATP using mitochondria, so that we don't have a lot of glucose in the blood, so that we don't get diabetes. We also need to make sure that we are always healthy, have a healthy diet, different types of nutrients, because we need to break down all these foods into the building blocks so that we can use it to make things for our own use. Like for example, we need to make enzymes in our body. Enzymes can be used, enzyme is a type of protein, and protein is made out of amino acid, and we need amino acids from other source of food that has protein. So let's say milk has lots of protein. We drink them and we break down milk into sugar and amino acid. Then we use these amino acids to make protein, human protein, that can act as enzyme, transport protein, antibody, and what's the major function? Structure, right? It can help us to repair our muscles. That's the idea behind it. So, with that, I conclude our Test 5 review from atoms to organisms. And we learned so much about different types of atoms, molecules, and how they work together for different systems, and so that you're alive. And after Test 5, after Easter break, we're gonna start learning the most important systems, how to make sure that your DNA can be passed to the next generation. Hopefully you enjoy our live show. If you have any question, feel free to ask me at the comment section. And Cheyenne, happy thanks for joining it, and whoever's watching this, thank you very much, and I wish you all the best in this coming test. Happy Snow Day.